we'll move on to the next heading, which is epigenetics. So as the name suggests, it is epi means above. So it is above genetics. So something above genetics is going to have a role over here. So these are actually heritable changes only. Heritable changes in the DNA which don't alter the DNA sequence. So heritable changes which are not going to affect the DNA sequence. So this is the exact definition of epigenetics. So what are these changes? These changes are going to be DNA methylation, then histone acetylation. So post translationally, it is going to alter the expression. So DNA methylation, I already told, it is going to cause silencing. So suppression of the gene expression. So silencing of the gene expression is going to be there by DNA methylation. So post transcriptionally, after the protein is being formed and translated, these can go and still alter the gene expression. So they're not altering the DNA sequence, they are just altering the expression by these changes. So this epigenetics is again controlled by various genes. So some examples alone I'll tell. So DNMT3A is going to control the DNA methylation. So DN is DNA and then MT is for methylation. So DNMT3A is going to be a gene controlling this epigenetics. So many of these epigenetic controlling genes are going to be mutated in leukemias and lymphomas. Okay. So DNMT3A is going to be mutated in acute myeloid leukemia, and MLL1 gene is again present, that is going to be mutated in acute leukemia, MLL2 in follicular lymphoma. So many such genes are going to be there. So leukemias and lymphomas are usually caused because of this epigenetic alterations. So now lastly, we'll see about microRNA. So microRNA, these are going to be non-coding RNAs. Initially also I mentioned non-coding RNAs like microRNA, long non-coding RNA, example of which was the ZIST gene, Lyon's hypothesis. Then we had transposons and all. So this microRNA is a non-coding RNA which has a very important role in regulating the gene expression again. So this is microRNA is going to be composed of 22 nucleotides. So 20 to 22 nucleotides is the length of this microRNA. They have a regulatory function which is basically inhibiting the translation of the mRNA. Okay. So microRNAs are again classified into two: oncogenic microRNAs and tumor suppressive microRNAs. So whenever there is some mutation in this microRNA and this oncogenic oncomers are going to be increased, it is going to result in oncogenesis. And when this tumor suppressor microRNAs are going to be decreased, they are going to again result in oncogenesis. So let's see the example of this oncomers and tumor suppressor mers. So oncomers example will be microRNA 155. So increased expression of this oncomer is going to cause upregulation of MYC. So MYC, we all know it is involved in lots of lymphomas, Burkitt lymphomas and all that, right? So it is going to result in B cell lymphomas. Another oncomer is microRNA 200. So microRNA 200, when this is increased, it is going to upregulated, it is going to cause epithelial mesenchymal transition. So epithelial mesenchymal transition is needed for the metastasis also, right? So for higher grade of tumor. So epithelial mesenchymal transition is associated with microRNA 200. So tumor suppressive microRNA example will be microRNA 15 and microRNA 16. So MIR15 and MIR16, when they are being downregulated, it is going to lead to upregulation of an anti-apoptotic protein which is BCL2. So this is actually seen in CLL. Okay, in CLL we are going to have upregulation of this BCL2 anti-apoptotic protein because there is going to be a deletion 13 in CLL. So in deletion 13q is seen in CLL. So this MIR15 and MIR16 is actually present at this at the 13th chromosome. So that part of the deletion 13q is going to cause deletion loss of this microRNA 15 and 16 as well. So that will result in upregulation of the BCL2, which is an anti-apoptotic protein. So there is going to be survival of the abnormal B cells. So that is the pathogenesis of CLL, which is chronic lymphocytic leukemia. Okay. There is something called as dicer mutation also. So during the formation of this microRNA, so microRNAs also need to be synthesized, right? So during their synthesis, there are going to be certain enzymes like DROSHA, then dicer. So these mutations can also result in diseases. So dicer is one such protein an enzyme which is needed for microRNA synthesis. So dicer mutations can also result in testicular and ovarian sex cord stromal tumors. Okay, sex cord stromal tumors are associated with this dicer mutations. So all of these are again MCQs. Okay, now we had seen about the firstly we started with the basics of genetics, then we saw about the various disorders classification, Mendelian, non-Mendelian disorders, then chromosomal disorders, multifactorial inheritance. Then we moved on to how to diagnose this lab diagnosis of this genetic disorders. Then finally we saw about this epigenetics and microRNA.